Okay, we are live on YouTube. Are both of you guys okay with your talks staying up as sort of in the archive?
Yep.
Awesome.
Yes, that's fine. Everything I'll show is on bioarchive as well. So
Awesome. Love it. Bioarchive's the best. I'm so glad at that invention becoming popular.
yeah.
It's really helpful. All right, welcome everybody who's joining. We'll just give this another 30 seconds or so, and then we'll get started. All right, cool. So we have people still coming in, but we'll go ahead and get started so we can keep to the time. So good morning or afternoon or evening to everyone. Thank you all so much for being here on our whateverth installment of cell migration seminars. I have to say, I feel like the world is just extra crazy these days and getting crazier by the day, and this weekly series has been like my stable rock. So I'm so grateful to you guys for presenting today and for everyone who's coming to listen. So really happy that we're doing this. So first, I think just to remind everyone, most of you guys have been here many times, but feel free to put your questions in the chat or just write that you have a question and we'll call on you if you want to unmute your mic. And we'll leave all questions to the end of the talks. So first up, we have Lotte de Winde, and she did her PhD at the Department of Tumor Immunology at the Radboud University Medical Center in the Netherlands on B-cell lymphoma development and dendritic cell migration. So Lotte has just finished up a postdoc in the lab of Dr. Sophie Acton, focusing on the role of Podoplanin in fibroblastic reticular cells and in amoeboid invasion of melanoma. And the latter project is what she'll be talking about today. And then she's actually just started a new postdoc in the MEBIUS group at Amsterdam UMC, where she will be studying how lymphoid stromal cells control anti-tumor immune responses. So if you want to go ahead and start sharing your screen, and the floor is yours. Thank you so much.
Thanks, Jennifer and Adam, for inviting me to present to you all today. So yes, today I will share one of the stories I've been working on while being a postdoc in the lab of Sophie Acton. About the role of Podoplanin in driving dedifferentiation and amoeboid invasion of melanoma. So cancer metastasis is a multi-step process. And from the primary tumor, the cancer cells need to get into an invasive mode to invade through the basement membrane and the surrounding stromal cells to then migrate into the bloodstream and find another niche to metastasize and develop proliferate and develop a tumor, a secondary tumor. So for today, we will be focusing on this initial first step of this invasion of primary tumor cells through the basement membrane and into the surrounding stromal cells later. So as you all know, there are many different modes of cell migration. And for tumor cells, they can either invade more collectively or as single cells. And in single cells, they can either invade as have a mesenchymal phenotype, but also some tumor cells develop an amoeboid phenotype, and that can either be more filopodia protrusions or really blabbing protrusions. And also tumor cells can switch between these different modes during their lifetime and during this process of invasion. But for today's talk, the first one depicted here, the amoeboid blabby form of migration will be the topic. So amoeboid blabbing migration is characterized by high contractility of the ectomyosin network. And this high contractility is ROC dependent, and there is high activity of RhoA and myosin 2. In amoeboid invasion, this blabby phenotype, the cells are very low adherent to their surrounding, and the contractile forces of the ectomyosin contractility will drive hydrostatic pressure will drive this blabbing protrusions, in which you then get new F-actin polymerization. And really, this is how these cells squeeze through to the extracellular matrix. And they really don't need proteases to do that. So this is a proteolytic independent mode of migration. So one tumor type or tumor cell that is well known for having this amoeboid cell invasion is melanoma. And a lot of melanomas have BRAF mutations, and for this type of BRAF mutated melanoma tumors, there are very good BRAF inhibitors on the market, and they are given to patients. However, not all cells in such a tumor are react to these BRAF inhibitors. So there are also BRAF inhibitor tolerant cells, and these cells will then just stay there, they won't die, and they will just grow, and they can obtain this amoeboid invasive phenotype and metastasize then very easily. So for this invasion, to target this invasive phenotype, because that's not being done by those BRAF inhibitors, there's a new pool of therapeutics on the market called microstatics. And these microstatics are basically inhibitors of this RhoA and ROC, and to inhibit this high contractile phenotype of these invasive cancer cells. However, we don't really know at what point, in what phenotype these tumors are. So there is a need for linking biomarkers to high contractile phenotype, and to define biomarkers to this amoeboid phenotype. And maybe also, if these biomarkers are linked to a phenotype, we can maybe also find targets on the outer surface of those tumor cells that are more easily targetable than intracellular proteins. So this is the idea we came from, where we started from, and in the Acton lab, we are interested in the protein Podoplanin, which is a glycoprotein. And a lot of our research is being done on Podoplanin on lymph node fibroblasts, which highly express Podoplanin. And in these cells, Sophie had found that Podoplanin is really linked to high ectomyosin contractility. So, and because we know that podoplanin is also very highly expressed on cancer-associated fibroblasts in tumors, but also on the tumor cells themselves, we hypothesized if maybe podoplanin in tumor cells also drives contractility. And we, as a model system here, we chose melanoma cells. And we believe, or we hypothesize that then this high podoplanin-dependent high contractility drives this amoeboid invasion of melanoma. So first we wanted to see if indeed in human melanoma we could find, or we wanted to determine the podoplanin status. So for this we used 2 cell lines that are actually derived from the same patient. So one is WM983A, which is derived from the primary melanoma, and the WM983B is derived from the lymph node metastasis in this patient. And if you look just at these cells under the microscope stained for F-actin, we can already see that this cell line B, so the metastasis cell line, has a very round phenotype. And this is also indicated in this quantification here, whereas the cell line A has a much more mesenchymal phenotype and is less round. And this is actually correlated with higher podoplanin mRNA expression in this cell line B, which has about 2.5-fold higher expression of podoplanin mRNA. And to confirm that this, because these are cell lines, that this is also seen in primary tumor samples, we looked into 2 data sets of primary melanoma with control samples, non-tumorous control samples. And in both data sets, we found that there is a significant or a trend towards a higher podoplanin expression in primary melanoma compared to benign nevi or normal skin on the right. So this indicates that indeed, at least in a subset of melanoma in humans, patients, we can find increased podoplanin expression. So for the rest of our study, we then switched to murine melanoma cell lines because that's a bit easier to work with. And we first looked at podoplanin expression in different murine melanoma cell lines. And the star with the cell line name indicates that these are BRAF mutant cell lines. But also here we have a cell line with low podoplanin expression, one with very high podoplanin expression, and also an intermediate cell line, the B16F10, which is a BRAF mutant non-BRAF mutated melanoma cell line. And actually this is the one we use because it has quite high podoplanin expression, but not as high as this 4434, which is very contractile, and these cells are almost rounded already in cell culture, so these are a bit more difficult to work with. So we use this B16F10 cell line to knock out podoplanin. So we could compare podoplanin positive with podoplanin knockout cells within the same experiment. And therefore we also gave them a different color. So we gave the podoplanin positive cells M orange and podoplanin knockout CFP. And this will be the same for all the data I will show you today. So first we looked in vitro at these cells in 2D, and we could immediately see that in the podoplanin knockout melanoma cell lines, that they were phenotypically completely different compared to the podoplanin expressing cell lines, which show a very rounded phenotype, and high F-actin, cortical actin, and they were also very clustered. Whereas the podoplanin knockout cell lines were much bigger, much less F-actin, and also they formed all these protrusions. And that we could also quantify that really the cell area and the perimeter of podoplanin knockout cells in vitro was much higher. But what happens when we cultured these cells together in a tumor? So we mixed the podoplanin positive and podoplanin knockout cells together and injected them in a mouse to form a tumor. And in these mixed tumors, we could actually see the same as what we saw in vitro. So we see that in areas where we have high podoplanin expression, or the podoplanin positive cells, that we see a high F-actin and cortical actin expression, which is even more pronounced in the zooms. Whereas in the cells that are podoplanin knockout, for example, we hardly see any F-actin. And also here we could really see that the podoplanin positive cells have a rounded morphology, whereas the podoplanin knockout cells really make these tiny protrusions as they were doing in vitro. So this data suggests that podoplanin promotes the contractility of melanoma cells in vivo. And really also here we could see that the cross-sectional area of the podoplanin knockout cells is higher than the podoplanin positive cells. So, but what does this mean for maybe the invasive capacity of these cells? So in this experiment, we took whole tumor sections and computationally analyzed the enrichment of podoplanin positive cells in the invasive front of these mixed tumors. So for each tumor, we identified a couple of invasive front regions and determined the enrichment score. And if the enrichment score is above 1, it means that this area of the invasive front is enriched for podoplanin positive cells, whereas if the enrichment score is below 1, it means it's enriched for podoplanin knockout cells. And actually in the 7 tumor sections that we analyzed, we could find 5 tumor sections where we saw an enrichment score higher than 1, meaning that in 5 out of 7 tumor sections in the invasive front, the podoplanin positive cells were enriched compared to the podoplanin knockout cells. And also when we combine these all together, we see that we get overall the podoplanin positive cells are more enriched in the invasive front. But enriched in the invasive front doesn't necessarily mean that they're also invasive. So we took a closer look at these invasive fronts and found that the cells that migrate into the surrounding podoplanin high stromal area were actually rounded podoplanin positive cells. And we really saw these single invading cells as a characteristic for this amoeboid cell migration, and we couldn't detect any podoplanin knockout cell invading into the surrounding stromal area. So this suggests that really this high podoplanin drives high contractility, and that then promotes this single amoeboid-like invasion of melanoma cells. However, is podoplanin expression sufficient to drive this? So for this, we turned it around and we took a podoplanin low cell line, which is the 555M cherry cell line. And part of these cells, these 55M cherry cells, we induced with podoplanin CFP to make them express podoplanin. We injected this mix of the low podoplanin expression and high podoplanin expression cells. We injected them in a mouse to form a tumor, and we imaged them with intravital imaging. And in this movie, you can already see that the green podoplanin expressing cells are really moving much more, whereas the 55M cherry cells are more wiggling, which is also due to the intravital imaging. So, and when we then take to look at this more statically and take 2 examples of cells, we can really see that these cells are making protrusions and moving, are really moving within the tumor. And if we then analyze that for all the cells in this tumor, we can see that there is a portion of static cells, which are mainly the just 5,5 mCherry expressing cells, whereas the Podoplanin CFP expressing 5,5 mCherry cells are really much more motile and their velocity is much higher. And then we also have a small portion of really migrating cells, so with a much higher velocity. And this is just only those Podoplanin CFP positive cells, because those cells also have the 5,5 mCherry. And here is where the two graphs really overlap. So only the Podoplanin expressing cells are really having these high velocity and therefore are really migrating cells. So this suggests that really Podoplanin expression itself, inducing Podoplanin expression in a melanoma cell line is sufficient to induce increased motility of these cells in vivo. And then to have a closer look at what happens when we inject Podoplanin into the cell line, on the left, you can see the 5,5 low Podoplanin low expressing cell line, which has this mesenchymal phenotype with nice protrusions. But if we give Podoplanin to this cell line, it really becomes a blabbing, moving, pushing away the collagen around it cell. And I also have two stills where you can really see that the high Podoplanin expression is really on the membrane of these cells, also when they make the blebs. So for a melanoma cell to become motile and invasive, it's actually a dedifferentiation program, because melanoma develops from melanocytes. But these melanocytes during development were migratory cells. So from the neural tube, melanoblasts migrate all the way to the skin, and there they will differentiate to become non-migratory melanocytes. So actually, during melanoma development, melanocytes become dedifferentiate and lose classical melanocyte features like pigmentation. So we looked in our cell lines if we could see something like that too. And strikingly, when we injected single tumors, which were only Podoplanin positive cells or Podoplanin knockout cells, we could see that those tumors from the Podoplanin knockout cells were completely black and pigmented. Whereas the Podoplanin positive tumors, we couldn't find this pigmentation. And this was actually cell intrinsic, because if we just pellet the cell suspension, we can already see that the Podoplanin knockout cells are really pigmented. And also in a mixed culture of Podoplanin positive and Podoplanin knockout cells, we can really see that these CFP positive Podoplanin knockout cells have these pigmentation and black granules in their cytoplasm. So pigmentation is linked to expression of certain genes. So we looked in the two data sets, data sets of primary melanoma and melanoma cell lines, if we could correlate Podoplanin expression to expression of these melanocytes and pigmentation genes. And interestingly, in primary melanoma, highly expressing Podoplanin, we see low expression of genes linked to pigmentation, like PMAL and tyrosinase, both in the primary melanoma, but also in the melanoma cell lines. And of course, then we also looked at the gene expression of these genes in our Podoplanin positive and Podoplanin knockout cells. And here you see the expression ratio of Podoplanin positive cells versus Podoplanin knockout. So Podoplanin knockout is expression level is set at 1. And we can see that melanocyte associated genes, so that's the non-migratory pigmented genes or linked to that phenotype, are low in podoplanin positive cells, whereas MIT-F and PU3F, which codes for BRN2, which is linked to a dedifferentiation of melanoma, are significantly higher expressed in podoplanin positive melanoma cells compared to podoplanin knockout cells. So in summary, I've shown you today that there is a subset of melanoma that increases expression of podoplanin in the primary tumor. And this is linked to a more dedifferentiated subset of melanoma, linked to a more invasive phenotype. And podoplanin as such can serve as a biomarker to determine the invasive capacity of melanoma tumors, and also when the doctor should maybe prescribe microstatics. However, podoplanin itself can also be a therapeutic target, because if we could mimic the binding of CLEC2, which inhibits this podoplanin-driven contractility, using an anti-podoplanin antibody, we could maybe revert the high contractility phenotype of high podoplanin expressing tumor cells, and as such inhibit their invasive capacity or their protection by CLEC2 expressing platelets in the bloodstream. So they can, when they come in the bloodstream, they die and they cannot migrate into a secondary site. So with that, I would like to thank the amazing Acton Lab, at which I worked with a lot of pleasure for the past 3.5 years. All my colleagues and everyone who has worked with me during these 3.5 years on this and other projects. Our collaborators on this project, Vicky, Samantha, who was first in the Acton Lab and now is a PhD student in Vicky's lab, and Eva for all the data set analysis, our funders. And this story is, you can find it and read it on bioRxiv. And I would also like to thank Ankita Jha for writing a really nice pre-lights on our preprint. And thank you very much for your attention.
Thank you. That was really great. I have a couple questions, but I see that there's similar ones in the chat. So we'll start with everybody else first and see what we can get through. Our first one's from Stephanie Wu. Maybe Stephanie, do you want to just go ahead and ask yourself since I know you're here?
So this is related to your in vivo experiments. Did you see that the podoplanin positive cells were migrating collectively?
You mean in the intravital imaging?
Mm-hmm.
Yeah. No, these were very short movies. So the movie I showed you was a total of 30 minutes. So, but we mainly saw really single cells moving in these tumors. But yeah, but they're very short, and also they're of course mixed with non-podoplanin expressing cells. So yeah, it's hard to say if they're moving, if they can move collectively. We haven't observed it.
Thanks. Gareth Jones asked, did you quantify the increased podoplanin surface expression in the podoplanin positive cells? And how much more do you need to affect cell morphology?
Yeah, that's a very good question. We had some technical difficulties with finding good podoplanin antibodies, and I don't have a clear answer for this. It looked like the difference in the WM cells, because that's the only one we looked at, the level of podoplanin expression on the surface was not very different, but there's maybe a difference in, I won't say clustering because it's just normal confocal microscopy, but localization of podoplanin on those cells. But we haven't analyzed that in depth, so I won't draw too big conclusions on that.
Great, thanks. From David Crickley, and I have a similar question. So what are the extracellular ligands for Podoplanin, and how does it signal to rock?
Yeah, so let me just quickly go out of my presentation. So the extracellular ligands for Podoplanin, the only one that's known is CLEC2. So that's a beta-galactan receptor expressed on platelets and on activated immune cells like dendritic cells. So in steady state, when it's not bound to a ligand, Podoplanin is linked to the actin site skeleton via ERM proteins, and that then leads to activation or GTP activation of RhoA, ROCK, phosphomycin light chain, which then drives this actomycin contractility. And when we know that when CLEC2 binds Podoplanin, that we lose phosphorylation of ERM. And then we lose this, we also get a shift from RhoA to RAC1. We lose phosphomycin light chain and this is all in lymph node fibroblasts, but we have not looked at this in the melanoma cells, but I assume it would be the same because it has been shown by others in tumor cell lines that there is a similar cascade going on.
Thanks. And we've got a question from Si Chang, who's asked, if you overexpress Podoplanin in fibroblasts instead of in melanoma cell lines, do you also see around morphology and blood formation? And a second question, do you know how Podoplanin controls branched versus linear actin bundles?
For the first question, yes. Expressing Podoplanin CFP into our lymph node fibroblasts makes them from very elongated cells into rounded contractile cells. The second one, we haven't looked at it, and I can't recall anyone has looked at it. So I'm afraid I don't have an answer for that.
Just a quick one from me. My understanding, and this is not my area, but my understanding was that melanoma is one of these cancers where it seems like EMT is playing a really important role. So I was wondering if there's any, interaction between the Podoplanin and sort of your EMT pathways.
Good question. I don't know, actually. Maybe other people have looked at this, but I don't know it by heart right now.
And I could be wrong about the link there. I might be confusing it with something else.
Well, I mean, in every tumor and also in melanoma, there will be not all invasive cells will be amoeboid. So there will also be mesenchymal cells. But I'm also not an expert on EMT, so I don't really know how that works in melanoma. And if there is a link with Podoplanin. Sorry.
No, thank you.
Thanks. Maybe we can make it the last quick question. So one from Giorgio Schitta, who says, nice talk. If I remember correctly, Gerard Cristofori group showed that Podoplanin expression in breast cancer promotes collective migration by increasing filopodia and reducing Rho GTPase activity. Are these effects epithelial specific, or more generally, do you envision a context-specific role for Podoplanin?
Short answer, I don't know. I will look up the paper and maybe then, sorry, I'm not familiar with the paper, so I will have to look. But I think that, we study Podoplanin, but of course in a tumor, there's so much more going on. So it could well be that there are other players playing a role. Podoplanin can also bind, which is indicated in this diagram, and I haven't talked about it, but it has other binding partners on the same cell. So Podoplanin can bind to CD44, it can bind to CD9, And in these interactions with its binding partners and with different binding partners, it does different things, which we are looking, or I've looked at on lymph node fibroblasts. So it could well be that in different tumors, Podoplanin maybe interacts with different binding partners, and as such has a different function.
Thanks again. Thanks a lot, Lotte, for a very nice talk.
Thank you.
I think we'll move on to the second speaker now. So our second speaker is Stephanie Wu. Stephanie is an assistant professor at the University of California, Merced. In her PhD and postdoc work, she worked on integrin dynamics during axonal guidance, morphogenesis of the heart and endoderm in zebrafish, and developed a zebrafish optimized optogenetic gene expression system. And now she's got her own lab, which investigates the cell biological mechanisms underlying tissue morphogenesis. And she'll be talking about the early zebrafish endoderm as a model of mesenchymal-to-epithelial transitions.
Great. Thanks, everybody. This has been a really great seminar series, so I'm really happy to be a part of it. Telling you guys about some of the work that we've been doing in my lab at UC Merced. So Lotte has already introduced the concept of different modes of cell migration. And my lab is a developmental biology lab. So what motivates us is, or one of the things that motivates us is this idea that dynamic cell behaviors like cell motility and cell migration can be thought of as a reflection of a cell's particular developmental state or cellular identity. So as an illustration of that, I have on this slide 3 different cell types that at some point in their developmental history exhibit a migratory phenotype. But these 3 different cell types exhibit very different modes of cell migration. So on the left, we have a spinal neuron growth cone that exhibits highly persistent directional migration. And that's in contrast to these endodermal cells in the middle here, which are the cells I'll be talking about. They exhibit highly dynamic but directionally random migration. And then finally over here on the right, we have early cardiomyocytes that exhibit collective migration. So the broad questions that my lab is interested in are, 1, how do the same developmental programs that specify these different cell types, how is that specification translated into these dynamic behaviors that are very characteristic and distinctive of these different cell types? And secondly, what is the significance of, for example, different modes of migration to the morphogenesis of that particular tissue, or even to the overall shaping of the embryo? So another way to say this is we're interested in both the how and the why of developmentally regulated cell migration. So to better understand these questions, we use the early zebrafish endoderm as a model system because this is a tissue in which the cells undergo several different transitions in the mode that they migrate in during their development. So the endoderm is one of the 3 primary germ layers that's formed early during embryogenesis. It's the layer that will eventually give rise to the epithelial lining of your gastrointestinal and respiratory systems, as well as other structures such as the pharyngeal arches and the thyroid. So in the zebrafish, endoderm is specified by about 5 hours post-fertilization. At this stage, the embryo is organized as sort of a ski cap of cells that sits on top of a giant ball of yolk. And it's these cells, this band of cells right here, right at the margin between the embryo proper and the yolk, that becomes specified as endoderm. So soon after endoderm specification, the embryo undergoes a process called gastrulation, in which the endodermal cells will internalize or ingress underneath their mesoderm and ectodermal neighbors, forming an inner layer of the embryo. However, during this ingression process, the endodermal cells actually lose adhesion between each other. So this inner layer that they form is actually not a coherent cell layer. So this has some characteristics in common with epithelial to mesenchymal transitions, although this isn't a true EMT because these cells were not mature epithelial cells to begin with. However, so during this early gastrulation stage, these cells exhibit highly dynamic movement, which you can see in these movies here. But at this stage, this migration is highly directionally random. So this has the effect of dispersing the cells all across that inner surface of the yolk in this sort of what we say a salt and pepper distribution. However, at the end of gastrulation, these cells undergo a switch in their migration mode and become highly directionally persistent, gathering at the dorsal side of the embryo, where they will then start to re-adhere to each other, eventually differentiating into a mature epithelium, which you can see down here through the asymmetric localization of ZO1. So this latter phase of endoderm development is more akin to a mesenchymal to epithelial transition. So it's how these different transitions are developmentally regulated that my lab is currently interested in. So to better understand the mechanisms that underlie these different transitions, I generated a transgenic zebrafish line that fluorescently labels the actin cytoskeleton specifically in these endodermal cells. This transgene consists of the endodermal SOX17 promoter driving expression of a GFP-tagged actin binding protein called eutrophin. So I hope you can see that this transgene really nicely labels actin-dependent processes like lamellipodium filoporeal protrusions, retrograde flow, and contraction of actin filaments. So using this transgenic line, we initially looked at this transition from directionally random to directionally persistent migration. And we determined that the early directionally random phase of migration is associated with increased actin dynamics. So we infer that in a couple of different ways shown here. First, we quantified the lifetime or the stability of actin-rich lamellipodial protrusions and found that they were somewhat labile during the early random migration phase, but then become stabilized as cells transition to persistent migration. We also, from similar time-lapse movies, generated chymographs from which we could measure the rate of retrograde flow. And we found that retrograde flow significantly slows again during the transition from random to persistent migration. So we next wanted to better understand the signaling pathways that could be regulating these differences in actin dynamics. So we initially focused on a signaling molecule called nodal. Nodal is a member of the TGF-beta family of signaling proteins that has a really well-known role in specifying endodermal fate. So we know that it's a signal that these cells have recently seen. We did a screen looking for cytoskeletal associated genes that could be transcriptionally regulated by nodal signaling. And we identified this gene called PREX1, which is a guanine nucleotide exchange factor, or GEF, specific for RAC GTPase. So RAC, of course, has a very well-known role in regulating actin dynamics. So PREX itself, its expression appears to be nodal dependent. So if we treat embryos with a pharmacological inhibitor of nodal signaling, called SB505-124, or SB for short, we see a downregulation of PREX expression. But when we overexpress a constitutively active nodal receptor, we see an upregulation of PREX expression. Consistent with the role of PREX as a RAC GEF, we used a fluorescent activity, a fluorescent reporter of RAC activity. And we found that, again, if we treated endodermal cells with this nodal inhibitor compound, we saw a strong reduction in the levels of RAC activity in these cells. Finally, we found that if we interfered with either nodal signaling with the SV compound or with RAC activity through expression of a dominant negative RAC, we could precociously switch these cells' migration mode from directionally random to directionally persistent, suggesting that this signaling pathway going from nodal to PREX to RAC activation is responsible for this random migration phenotype that's characteristic of this particular stage of endoderm development. So we next wanted to ask what the significance of this random migration phase is to the overall development of the endoderm. So again, we're not only interested in how migration modes are regulated, but also why. So to do this, we took advantage of the fact that expressing dominant negative RAC in endodermal cells caused them to precociously exhibit directionally persistent migration, skipping that random migration phase altogether. So we took endodermal cells expressing dominant negative RAC, mixed them together with control endodermal cells, then transplanted them to unlabeled host zebrafish embryos, and then just asked what happens to these cells over time. So this is a zebrafish embryo at about 24 hours post-fertilization. And we find that the control endodermal cells in red tended to end up in endodermally derived tissues where we would expect them to be, so places like the lining of the gut tube. In contrast, these cells expressing dominant negative RAC contributed to both endodermal as well as non-endodermal tissues, including the muscle and the notochord. So suggesting that forcing cells to bypass this random migration phase interfered with the maintenance of their endodermal identity. So now, with a better understanding of how this phase of endodermal migration is regulated, we want to next understand the transition. Excuse me, the lights turned off in my office. Okay. So we next better wanted to understand the transition between this directionally random sort of single cell-like migration to the convergence of cells into an endodermal sheet. So, so the current projects in my lab are really focused on this transition space. And we have a set of projects, and we're looking at this from multiple different angles. We have a set of projects that are interested in the changes in cell mechanics during this transition. We're also very interested in how cell-cell adhesions are built, such as adherence junctions, how these are built in vivo as the cells are differentiating during this process. But in the interest of time, I'll tell you about one project that we're looking at, trying to understand how cells are changing their interactions between each other over time. So we know from our time-lapse analysis that the transition from single cell directionally random migration to convergence into an epithelial sheet occurs gradually and progressively over time. So this is, again, using our fluorescent actin recorder line, looking at these movies are taken from the same embryo at different points in time. And what we can see is during early stages, cells occasionally make transient contacts with each other, but they're very short-lived. If we look, however, about an hour later, we can see that, again, cells make contact with each other that lasts for a little bit longer, but they still pull apart eventually. However, eventually, you can see these two cells here are flush with each other, and this third cell will come in and join them. So we like to call this the romantic comedy model of sheet formation. And we can see this when we quantify the duration of contact between cells at sort of early stages versus late stages. And we can see that they remain in longer contact with each other over time. So this progressive increase in cell contact and cell adhesion can be partially explained through the increase in expression of adhesion molecules such as E-cadherin. But one of the things that really caught our eye is a process that you can best see in this middle movie here, where when cells come into contact with each other, they appear to lose actin content specifically at that site of contact. And so that was reminiscent to us of this process called contact repulsion, or sometimes also referred to as contact inhibition of locomotion. So this occurs when two cells collide with each other and they experience a loss of actin at the site of contact, coupled with rapid membrane retraction of those contacting sites. And this is also accompanied by a repolarization of the cell with new actin polarization on a contact-free edge of the cell, which then results in the two cells pushing away from each other. So during the early phase of endoderm migration, so the phase that's characterized by directionally random migration, we also can see several examples of cells colliding with each other and then apparently repelling away. So, so contact repulsion has been described in many different cell types, and there have been several different signaling pathways that have been determined to mediate contact repulsion. The one common feature of almost all of these signaling pathways is the activation of the Rho GTPase Rho A. And Rho A seems to be pretty critical for orchestrating the major events that characterize contact repulsion. So of course, we then wanted to know whether the repulsive behaviors that we see in our endodermal cells, if that is also dependent on Rho A. So we overexpress a dominant negative version of Rho A in these endodermal cells and found that when they collide together, rather than repelling away, they seem to just slide right past each other. We can also see this if we quantify the collision angle, and we found that cells expressing dominant negative Rho A did not appear to change their migration direction in response to that collision event. So then we wanted to ask, what is the significance of this contact repulsion behavior on overall endoderm development? And so we looked at embryos at the end of gastrulation. So this is a stage at which the endodermal cells are maximally dispersed across that inner surface of the embryo. But not only are they widely dispersed, but they exhibit this very even, regular spacing between each other. And it's this spacing regularity that appears to be disrupted when we express dominant negative Rho A and inhibit and block that repulsive behavior. So in these embryos, we see large gaps that appear to be devoid of cells, interspersed with irregular clusters of endodermal cells. We can quantify this through a regularity index, which is a measure of the variance between nearest neighbor distances between endodermal cells. And we found that there was a significant decrease in spacing regularity in embryos expressing dominant negative Rho A. So this leads us to a model whereby these repulsive interactions between endodermal cells early on help to achieve this regular spacing between them. Interestingly, this mechanism of spacing regularity by repulsive interactions is often seen in the nervous system. So things like topographic mapping, positioning of the neuronal cell body, or dendritic tiling. And I bring this up because at this point, we still don't know the identity of the cell surface molecules that mediate interactions between endodermal cells. But we did do transcriptomic profiling of these cells at this stage. And interestingly, we found that genes related to axon guidance or axon pathfinding were overrepresented. So these include classical repulsive axon guidance cues like F-effrin, slit-robo, and semaphore and plexin signaling. So this is something that we are actively pursuing now in the lab. So if these cells are early on expressing repulsive interactions, we initially thought, okay, so to transition to convergence and sheet formation, you just turn that repulsion pathway off. But it turns out that things may not be that simple. So again, using our fluorescent actin line, we looked at cells specifically during the process of sheet formation. And we found that, yes, although cells are now adhered to each other rather than repelling away, we still see preferential actin polymerization at what are apparently cell-free edges. So you can see that in this movie here, as well as in these still images here. So that gives us this intriguing possibility that perhaps at this later stage, when cells are undergoing sheet formation, they may be partially co-opting this contact repulsion pathway as a mechanism for them to recognize cell-free edges, protrude into those cell-free edges, and ensure coherence of that sheet that they're trying to form. So because we previously determined that Rho A is required for these repulsion behaviors, one way that we could test this would be to interfere with Rho A signaling specifically at this late stage while leaving things unperturbed at earlier stages. So to do that, we are currently taking advantage of an optogenetic gene expression system that we're also developing in our lab. This expression system consists of a light-activated transcription factor called TAIL that in the presence of blue light dimerizes and binds to a regulatory element called C120, activating expression of a gene of interest. So using this TAIL system, you can control induction of your gene of interest both spatially and temporally by patterning the illumination of activating blue light. Or alternatively, you can control expression of this light-activated transcription factor with a tissue-specific promoter such as SOX17, and then deliver activating blue light using a simple LED panel. So this expression system was initially developed during my postdoc, but in my lab, we've continued to modify the system, and we've just recently posted a preprint describing an improved version of the system called TAIL 2.0. So current projects are, so currently in the lab, we're using the TAIL 2.0 system to induce expression of dominant negative Rho A and really test our model of the role of contact repulsion. Okay. So with that, I'd just like to thank a few people. So some of this work had, so early parts of this work were done, were based off of my postdoctoral work with Didier Stanier and Orion Weiner at UCSF. The TAIL 2.0 project was a collaboration with Stefan Materna here at UC Merced, along with Kevin Gartner and his previous graduate student, Laura Mota. Jessalyn LaBelle was also working on the improvements to the TAIL system. She's also now currently working on the contact repulsion project. And former members Adela Ramos and Kyle Shen also contributed to the TAIL 2.0 project. These are my funding sources, and I'm happy to take questions.
Thanks a lot, Stephanie.
Thanks. Thanks a lot. I was just curious if you had any thoughts about, so I'm biased, but any thoughts about this idea of a jamming transition? Like, do you think that there's anything that you could approach just sort of from a physical perspective of as these cells are sort of getting more crowded and getting more densely packed, if there might be some physical rigidity transition or something going on there that could be contributing to the phenomenon you're seeing?
Yeah, no, that is a really great question, and it's definitely something that we're interested in, because yes, you're right. As they're converging together, they're also becoming more crowded. So we're interested in both. So in terms of sort of mechanical changes, we're interested in both, like whether there are sort of intrinsic changes to the cell mechanics, as well as these sort of non-autonomous changes. And the other big unknown is what is happening to the matrix that presumably they're migrating through at this time. So
yeah, that was my other question, right? Because we see them as these little isolated, but we know that they're in a 3D environment, right? They're moving through something. So
yeah.
Do you know is it known what's surrounding the cells?
So they're migrating on top of the yolk surface, which I think is primarily fibronectin. But yeah, but what the architecture of that matrix is, we don't really know.
Yeah, I was just looking at your images. I was reminded of some work from the Heisenberg group where they've talked about the sort of like the idea of a percolation network where, once you reach sort of a critical density, then the forces can be transmitted through the cells.
Right.
And, yeah, cool. Okay, sorry, we'll move on to other people's questions. Let's see, from Ankita Jha, do you think that persistence increases due to cell-cell adhesion, or is there some extracellular ligand gradient during the late stages?
Yeah, no. So the persistence, the change in persistence happens just right before the cells converge together. So we can detect an increase in persistence even when the cells are sort of separated from each other. And so work from other groups have shown that there is a chemokine signal, so CXCL12, that becomes expressed at these time points, and so most of us think that is the external cue that guides them to that particular place in the embryo.
Thanks. Question from Raphael Petrosian, who says, thank you for a very interesting talk. Could you elaborate more on the Rho-dependent contact repulsion mechanism? Namely, how does a cell feel or measure the distance from other cells?
Oh, yeah, that's a really good question. So part of that we can't really answer yet because we don't know the identity of the molecules, the proteins that the cells are using to actually recognize each other. I think once we have a better handle on that, we'll be able to answer that question in better detail.
Great, thank you. From Si Chang, do you think that the changes in cell fate in the dominant negative RAC cells are due to impaired migration and hence the wrong place these cells end up to the changed environment for differentiation?
Yeah, so we think in those experiments, by precociously undergoing persistent migration, these cells, so these cells end up on the dorsal side of the embryo sooner than they would normally. So we think that they're sort of accidentally being exposed to other cues, so presumably mesodermal cues that's sort of switching their or biasing their fate more towards a mesoderm fate than an endoderm fate. But that's just sort of speculation at this point.
Thanks. I was wondering more about the random part of the cell migration at the beginning. So that is causing the endodermal cells to maintain their fate, but is that independent from the randomness of the migration per se, or is that correlatory or causation?
I mean, I think it's the random migration, I think, is a result of them being specified as endoderm, and then I think the reason or the significance of it is to produce this dispersal effect to get them to be dispersed all across the embryo.
Okay, awesome. So this will be the last question, unless anyone else wants to jump in, go ahead and do that. So from Tony Tsai says, very cool work. I am surprised that the dominant negative RAC endoderm cell, while not migrating randomly, can enter persistent migration early. So does the dominant negative RAC also affect the persistent migration mode? Do you need to play with the dosage of dominant negative RAC to achieve this?
Yeah, hi Tony. Yes, we had to play around with the dosage quite a lot. So anybody who works with rare G2 phases know if you express too much of it, the cells just don't move at all. So yeah, we did have to play around with the concentrations to get it to a point where they would still move, but they moved in a more directional manner.
Okay, awesome. All right, well, I think that is all the questions that we have. So I just want to say thank you so much to both Lotte and Stephanie. So you guys, this was really, really interesting work, and thanks for being here. And then we'll be back next week. We have Hamid Badmos and Kyra Campbell. And so we will see you all